Hello students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia, Central University. Today we are going to talk on module conformation of protein, in which we discuss first the concentration measurement of the protein and then conformational changes in protein, and we especially discuss the protein melting under the different conditions under the paper techniques used in molecular biophysics part 2. After this lecture, the students will be able to understand overview of the protein conformation, what are the protein structure and how it is conformation. Then we discuss about the methods used for the protein concentration measurements. Then we talk about the protein conformation from synthesis to function, right from the synthesis of the protein in the cell and then how it achieves the function. Then we talk about the factors that determines the conformation of the protein, and at last we discuss about the protein denaturation and melting, especially methods and its significance. So we first discuss about the overview of the protein structure. As you all know that the protein are synthesized by the cellular machinery like ribosome, and the ribosome synthesizes the protein which is having information on the messenger RNA and gives a linear polypeptide chain of the protein, and this linear polypeptide chain is converted into a well-defined three-dimensional structure that's important for that is the essentially important for the function of the protein. So as they are synthesized, they assume secondary and tertiary structure like alpha helical conformation, and then they form a compact structure that's known as three-dimensional structure or tertiary structure. So the activity of protein is depending on the integrity of its final tertiary structure, and it's also referred as native form, because this form is generally exist in the cell, and loss of structural integrity is leads to the denaturation of the protein, and of course it is the activity is lost. So the main force that governs, that is responsible for the maintenance of three-dimensional structure of protein, is the hydrophobic interaction that contribute strongly to the protein folding and stabilization. We discuss about the journey of protein from polypeptide. You can see here the messenger RNA, which carries information about the protein sequences, bound to the ribosome and transfer RNA, and which lead to the synthesis of the protein in the linear form that is known as the unfolded state. From the unfolded state, it has to go into the many different cycles. It can be simply destructed by the cellular machinery, it's simply degraded by the cellular machinery, or it bind to the some chaperone molecules which converted into the fully active and the native conformation. If it is unable to do so, then it may form the aggregates and it may form amyloids. We discuss how the structural information of protein is hidden in its sequence. For that, a well-known scientist, Professor Anfinson, performed a very simple experiment. He took a model protein, ribonuclease A, that having the 6-cysteine residue forming the 8-cysteine residue forming 4 hydrogen bonds. So it is the native and catalytically active state. He added mercaptoethanol and urea to the ribonuclease A solution. The urea is used to denature the protein structure, and mercaptoethanol breaks the disulfide bond. So the protein becomes fully unfolded and catalytically inactive state with reduced disulfide bond. Then he further removes the mercaptoethanol and urea by simple dialysing, found that the bond formation of the disulfide bond formation is the same as it was in the native conformation, despite of 105 different combination of disulfide bond formation. Only a certain type of disulfide formation was allowed. So it's suggesting that the structural information of protein is hidden in its sequence. We discuss about the energy landscape of protein structure and its conformation. You can see that Simply, we discuss from the primary structure to the tertiary structure and how it is energetically favored. So you can see simply the amino acid. If you mix the two amino acid in a solution, you can see the amino group and carboxylic acid group of the two amino acid forms the covalent bond known as the peptide bond. And this peptide bond is partially double bond characteristic feature, and that's why it having the flattened structure, and thus it allows the peptide bond allows the rotation of C alpha atoms towards a plane, and that's what it regulates the tertiary secondary structure formation of the protein. You can see here in the middle panel that the primary structure is simply the sequences of the amino acid, linear sequence of the amino acid. Because of a structural information hidden in its sequence, it either form beta sheeted structure or the alpha helical conformation. Sometimes if there is no structure alpha and beta sheet, it means it is the random coiled structure, and thus the whole polypeptide chain enfolded into the well-defined three-dimensional structure. It is known as the tertiary structure of the protein. In few proteins, the many polypeptide chains are held together and form the quaternary structure of proteins, such as the well-known example is the hemoglobin. So if you see the energy dynamics from the primary structure to the tertiary structure, the unfolded state or the primary structure, it can be having a large number of conformation, and that's what it possesses high energy and high entropy. But due to the systematic arrangement, it leads to the formation of medium kind of energy and lesser number of conformation as compared to the unfolded state and some sort of the lesser entropy, and that's known as the multinuclear state. And final structure which having the minimum energy is known as the native conformation, which is having the minimum entropy, minimum energy, and maximum stability and functional state of the protein. We discuss about the protein estimation. The quantification of protein content is important and it has many clinical applications. There is a wide variety of the protein assay available, but each assay has its own advantage and limitation. So first we must know that what are the factors that we should consider before choosing a method. These factors are most important: sensitivity, the presence of interfering substances, and of course time availability of the assay. So a large number of methods are available to estimate the protein concentration, and different methods have their own advantage and disadvantage. These methods are biuret method, Bradford methods, Folatemetric method, Geldal methods, BCDA methods, UV visible spectrophotometry method, and Folatemetric method. You can see here the development of color is directly proportional to the concentration of protein that is used to estimate the concentration of protein in an unknown sample. We compare the different methods of the protein estimation. For example, you can see here the biuret method. It is the relatively having low sensitivity for 1 to 20 milligram of the protein is required, and it requires lesser time, 20 to 30 minutes, and for this reagent is alkaline copper sulfate, and it has a simple disadvantage of the destruction of protein sample. The other method is the Lodi method. It is the highly sensitive, it having the high sensitivity. It used to estimate up to 5 microgram, and it of course it requires some more time, 40 to 60 minutes, and it is based on the copper Folin's Catacho reagents, and destruction of the protein sample also occurs. The Bradford method is relatively highly sensitive. It can estimate up to 1 microgram of the protein and requires lesser time to the 15 minutes, and the reagent is very simple, comestible blue G225 G250. And the BCA method is also very sensitive, although it requires more time, but it is the highly specific method. Other method is the spectrophotometric method. It is the simple, but it requires large number, relatively high amount of the protein, and no reagent is required, but it is the more empirical methods. We discuss the steps of the Lodi method. You can see here that the first step is the reduction of copper ion under alkaline condition, which forms a complex with the peptide bond. And in the second, the reduction of the Folin reagent by the copper peptide bond complex, which subsequently causes a color change of the solution into blue with an absorption in the range of 650 to 750 nanometer. The advantage of this assay are sensitivity and most importantly accuracy. However, it requires some more time than the other assay. So you can see here there are two conformation of the protein. One is the natively one of the linear random coil structure. There is no structure at all. It is known as the unfolded state, and by the cellular machinery, by the cellular environment, it is converted into the well defined three-dimensional structure. It is called native conformation of the protein. So these two types of conformation of protein is the extreme end. One is the unfolded state, that's the simply synthesized protein from the messenger RNA, and then it converted to the fully folded three-dimensional structure, that is the active form of the protein and also termed as the native protein. For estimation of protein concentration, especially for the unknown protein concentration, we must have a standard curve. That standard curve is actually showing the linear relationship between the absorbance versus the concentration, and the amount of protein in the sample can be estimated by an standard curve for the selected protein. For example, we take the casein, we can take the well-known protein like the BSA, and the standard curve is a type of graph that is used for the quantitative measurements. A standard curve of protein concentration is often created using the known concentration of bovine serum albumin, and then this concentration curve is used to estimate the concentration of unknown sample. So the protein will analyze is bovine serum albumin, and it the serum protein that transports fatty acid from the and important for the pH of the plasma. The protein quantitization assay BSA serve as a reference protein and is used to construct the protein standard curve. Other protein can also be used depending on the physical and chemical properties of your protein of interest. So you can see here in the standard curve, you can see the absorbance is at the y-axis and the concentration of protein at the x-axis, and as you increase the concentration of protein, there is a linear curve. Now you can measure the optical density or absorbance value of the particular unknown sample, and then you can directly estimate the concentration, exact concentration of protein by putting the value of absorption in this curve, and then you can estimate the concentration of protein of interest. Now we discuss about the biuret test. It is the most commonly used method for determination of the total protein in a sample. The biuret method is based on the complexation of copper to the functional group of the protein peptide backbone. The protein backbone having a functional group like CO and NH that forms the bonding with the copper ion. And the formation of copper protein complex requires two peptide bonds and produces a violet colored chelate products, which is simply measured by the absorption spectroscopy at the 540 nanometer. So over a given concentration range, the measured absorption of 540 nanometer is linear with respect to the concentration of protein, and the intensity of color and hence the absorption at 540 nanometer is directly proportional to the protein concentration, and it strictly follows the Beer-Lambert law. The another method is the biuret method, in which the hydrated copper sulfate that provides the copper 2 ion which forms the chelate complexes, and copper 2 ion produce the characteristic blue color, in which the potassium hydroxide actually do not participate in reaction, but it only provides the alkaline medium. And the another important component is the sodium potassium tartrate. It stabilizes the chelate complex and prevent precipitation of the copper hydroxide and potassium to prevent auto reduction of the copper. You can see here clearly that how the copper is complex with the two polypeptide chain, and then you can see as you increase the concentration of protein, the intensity of blue color is increased dramatically. So that value, the intensity of color, is used to estimate the amount of protein in the unknown sample. We talk about the Bradford method, You can see here that as you increase the concentration of protein, the intensity of blue color is increased. For the Bradford method, we use Comasi Brilliant Blue G250 dye in a colorimetric reagent for the detection and quantification of total protein. In the acidic environment of the reagent bind to the Comasi dye and results in a spectral shift from the reddish brown of the dye, which has absorption maximum at 465 nanometer, to the blue form of the dye, which having the absorbance at the 610 nanometer. The development of color in Comasi dye is based on Bradford protein assay, has been associated with the presence of certain basic amino acid, primarily arginine, lysine, histidine in the protein. Free amino acid peptides and low molecular weight protein does not produce color in the Comasi dye. Unbound forms are green or red. The advantage of this method include that it is highly sensitive and is able to measure 1 to 20 microgram of protein, and method is very fast. You can see here the Bradford principle. This is the structure of the Comasi Brilliant Blue G250, which is earlier it is the brown in color, but binding with the protein it produces blue color, which is estimated at 95 nanometer, and the difference between the two form of dye is greatest at 595 nanometers. So that's the optimal wavelength to measure the blue color from the Comasi dye of the protein complex. We talks about the biciclinic acid, simply called BCA method. It is based on reduction of the cupric ion to cuprous ion by the proteins. BSA reagents and copper sulfate is the main component of this reaction, and the outcome of reaction is analyzed by measuring the absorbance at 562 nanometer. This is one of the most sensitive method and requires minimum amount of the protein. You can see here in the structure that how the two polypeptide chains are interact to the copper ion and forming the complex. You can see here in the A, BCA, copper plus 1 BCA, then in the B, BCA, copper plus NTBP, and in the third C panel you can see NTBP, copper plus NTBP, and this method is used for the estimation of protein at the minimal concentration. We discuss about the Jildal method. This method consists of three basic steps. First step is the digestion of sample in sulfuric acid with a catalyst, which results in the conversion of nitrogen to ammonia. The second step is the distillation of ammonia into a trapping solution, and the third step is the quantification of the ammonia by titration with the standard solution. The sum of the organic nitrogen, ammonia and ammonium is proportional to the protein concentration. This is the Jildal method, structure of the Jildal flask shown here, that the protein solution is boiled and the nitrogen is trapped in the solution, and how the nitrogen is estimated. The UV spectrophotometric method is the most commonly lab method for the estimation of protein concentration, in which you don't need any reagent. Simply put the protein in the cuvette and then you measure the optical density of the protein at 280 nanometer. It is because of the tyrosine and tryptophan content of the protein, as the amount of tyrosine and tryptophan content in the protein is directly proportional to the absorbance, and of course it is the used to estimate the concentration of the protein. At least extent, the phenylalanine and disulfide bond also contribute for the absorption value. For the experimental procedure, the optical density of the test solution is measured at 260 nanometer and 280 nanometer by using the following formula. Like, you can use the protein concentration milligram per ml equals to 1.55 into optical density at 280 nanometer minus 0.76 into optical density at 260 nanometer. The optical density of the protein solution is depending on the three chromophore, like you can see tryptophan, tyrosine and phenylalanine, in which you can see that this is the absorption spectrum of the all three amino acids in free solution. And you can see the contribution of tryptophan is the maximum because it having the two aromatic rings, while the tryptophan is a little bit lower, and phenylalanine is a very low. So but anyway, all these three chromophores is majorly responsible for the absorption of protein at 280 nanometer. We discuss folatemetric method. It is based on the derivatization of protein with O-thiolaldehyde OPA, which reacts with the primary amines of the protein. The sensitivity of the test can be increased by hydrolyzing the protein before it testing. You can see here the shift base formation between OPA and the glycine. We talk about the protein conformation from synthesis to function. You can see here that the protein synthesis occurred in a large number of steps. After the synthesis, it further requires a lot of cellular machinery such as the GROES system, which helps to the protein to correctly fold and then forms the fully well organized tertiary fold structure of the protein. Now we discuss about the molecular chaperone that's responsible for the folding of protein from the lean random coil structure to the three-dimensional structure in the living system, especially in the cellular environment. So the nascent polypeptide comes from the ribosome and the folded spontaneously. Molecular chaperones are involved in their folding in vivo and are related to heat shock protein known as HSP. Minimize heat and stress damage of the protein that's lead to the protein renaturation, or it's another way around degradation. The molecular chaperone facilitates the correct folding of protein by minimizing the aggregate formation or the other misfolding events. So it binds simply to the nascent polypeptide to prevent premature folding. And the molecular chaperones bind to nascent polypeptide, and then it facilitates the membrane translocation import by preventing folding prior to the membrane translocation. It also facilitates the assembly and disassembly of multi-protein complexes. We discuss the different types of forces that involved in the stabilization of three-dimensional structure of the protein. You can see here in the structure, these forces are hydrogen bond, which is formed between the oxygen and hydrogen atoms, which is attached to the electronegative elements such as itself oxygen on the nitrogen. Then you can see here the hydrophobic interaction. It is found in the interior of the protein, disulfide bonds and weak electrostatic interactions and some van der Waals interactions. So the disulfide bond stabilizes the native conformation of protein and most stable or one of the strongest bond. Then the hydrophobic interactions are present in the interior of the protein, and it provides the maximum stability of the protein because it helps in the collapsing the protein, maintaining in the protein the three-dimensional conformation. The hydrogen bonds pairs between the residue of the water and the polar groups within the protein, and some ionic interaction stabilizes the structure of the protein. We talks about the protein denaturation. Denaturation is a phenomena that involves transformation of well-defined folded structure of a protein formed under physiological condition to an unfolded state under the non-physiological conditions. It occurs suddenly and completely over a narrow range of condition, and sometimes it is reversible. You can see here in the structure that the structure of normal protein and the structure of denatured protein, and these two forms are reversible under certain conditions. Now we see that what are the consequences of the denaturation of the protein. The protein denaturation leads to the loss of enzymatic activity, destruction of the toxin properties, improved digestibility, loss of solubility, and change in the texture. Protein stability is defined as the net balance of the forces which determines whether a protein will be in its natively folded conformation or a denatured state, providing an understanding of the basic thermodynamics of the process of folding. The net stability of protein is defined as the difference between free energy of the native state and denatured state. Both free energy of native and unfolded state contribute to the value of Gibbs free energy, that is the delta G, which is defined as the minus RT ln K. where K is the equilibrium constant. We talks about the protein denaturants. This graph showing that how the balance between pressure and temperature maintains the three-dimensional structure of the protein. If you move either towards the higher side of the temperature or towards the lower side of the temperature, similarly lower towards sides of the pressure as well as the temperature, the protein got denatures. However, a wide range of the temperature and pressure maintains the three-dimensional structure of the protein as shown in the green color. There are several denaturants are used to denature the protein. These denaturants are heat, ultraviolet radiations, strong acids or bases, urea and guanidinium hydrochloride, some organic solvents such as ethanol and acetone, and agitation is also causing denaturation of the protein by shearing of hydrogen bonds. You can see the extremes, how the extremes of pH changes the three-dimensional structure of the protein and its conformation. High pH and low pH denatures many protein. However, some proteins are quite stable at pH 1. The basic idea is that the net charge of a protein due to titration of all the ionizing group leads to the intramolecular charge repulsion, which is sufficient to overcome the attractive forces, mostly hydrophobic and dispersive, resulting in the least partial unfolding of the protein. Now we talk about the pH-induced denaturation. The presence of a specific counterion binding leads to the formation of compact intermediate state, such as the molten globular state that have substantial secondary structure, little or no tertiary structure, and relatively compact as compared to the native state. And proteins are most stable in the vicinity of their isoelectric point. In general, the electrostatic interaction are believed to contribute to the small amount of the stability in the native state. However, there are maybe some exceptions. Chemical denaturants. Urea, especially usually at 8 molar, and guanidinium hydrochloride at 6 molar are currently the best sought for the preferential solvation of the denatured unfolded state, which involving predominantly hydrophobic related properties and to a lesser extent hydrogen bonding. Both side chain and backbone appear to be more soluble in the presence of denaturants. These are good solvent because of the hydrophobic component and bad for the hydrophilic ones, and vice versa. The M value of the both the denaturants reflects the dependence of free energy on the denaturant concentration. For the urea, it is approximately generally 1 kilocalorie per mole, and for the guanidinium hydrochloride, it is generally 3 kilocalorie per mole. You can see here the structure of urea and guanidinium hydrochloride. How the chemical denaturation occurs by the urea and guanidinium hydrochloride is shown here. You can see here that polypeptide backbone collapse without side chain, and side chain reduces backbone collapse. However, persistent backbone collapse without side chain and side chain prime backbone expansion. You can see here as you increase the urea or of course guanidine hydrochloride concentration, here I am showing the example of fluorescence. However, the absorption and some other techniques can also be used. So a dramatic change of the native state and the unfolded state can be observed. Here you can see here a simple sigmoidal curve is followed by increasing the urea concentration, and the value of the CM is considered as a concentration where 50% of the protein is in denatured state and 50% of the protein in the native conformation, and it gives the idea about the stability of the protein. As the CM value moves towards the lower side, means protein is less stable, and if the CM value moves towards the higher side, it means the protein is more stable. Here we discuss the thermal denaturation. You can see here in the graph that as you increase the temperature, the optical properties is increased, and this tendency is used to estimate the thermodynamic properties of the protein. And you can see how the structural changes or how the conformation of the protein changes as you increase the temperature, and how the protein is started unfolding from the lower to the higher temperature. So the effect of temperature on the structure can be directly used for the estimation of the Gibbs free energy which causes the disruption of hydrogen bonding and increasing hydrophobicity. There is a compromise between the stability and activity in the structure of the active state of the protein. As you increase the temperature, the activity get lost and the structure of course get lost. Here we give some idea about the cold denaturation of the protein. Some protein also denatured as it moves towards the lower temperature. You can see here the protein structure of protein at 25 degrees Celsius. As you move down till the minus 11 degrees C, you can see a dramatic loss in of the structure protein occurs. The free energy curve starts to drop at lower temperature as predicted by the thermodynamics of the protein folding. And several proteins have been shown to exhibit cold denaturation under destabilizing condition in usually either low pH or moderate denaturate concentrations. Here we discuss about the denaturation of the protein by organic solvent. You can see that the exposure of protein to an organic solvent such as alcohol may also cause the denaturation. The protein molecule twists and flexes through free bond rotation within as the hydrophobic side chain shuns the alcohol. In this process, hydrogen bonds and ionic interaction can be broken. You can see here that the alcohol and binds to the forms hydrogen bonds and thus breaks the hydrogen bond, intramolecular hydrogen bond of the protein, and thus causes the denaturation of the protein. Here we discuss the denaturation of protein by detergent. You can see that the detergents has a hydrophobic side chain, hydrophobics as well as the hydrophilic side chains. Protein have hydrophobic and hydrophilic side chain, and detergent is attracted to these and forces the protein part. The native structure of protein is partially created by hydrophobic and hydrophilic interactions. Detergents substitute this self-bonding with the detergent amino acid bonding. Detergent is a salt and breaks up positive and negative interaction and thus unfolds the protein. You can see here in a graph that the before SDS treatment and the after SDS treatment, how the structure of protein get disturbed from the native conformation to the random coil conformation. So you can see here as we increase the pH of the solution by adding the hydroxyl ion, you can see the charge state of the amino acid, especially the main chain and the side chains, are changing, and as you increase the hydroxyl ion concentration means increase the pH, the protonation deprotonation occurs and the charge state converted into the uncharged state, and that's what there is a loss of counter ions, and that leads to the loss of weak electrostatic interaction, and thus leads to the denaturation of the protein. The denatured state of protein is simply saying that it involves the most of the residue in fully extended conformation of the peptide. So that's what it is able to highly exposed to the solvent, and thus the substitution involves solvents, and exposed residue in the native have the limited effect. On the other hand, the denatured state have considerable residual structure. Then it is also possible for the mutation and may affect the conformation of the three-dimensional structure and the unfolding state. In extreme cases, perhaps only the denatured state and is not in the native state. So you can see here the denatured state is simply the random coil state, and it have a large number of conformation. There is no net exact conformation of the protein. That's what it is called the unfolded state or the denatured state. Now we can summarize that as you increase the concentration of protein, the intensity of color development can be observed, and that is simply used to estimate the concentration of protein in an unknown solution. Now you can see here that there are several forces, non-covalent and as well as covalent forces, that stabilizes the three-dimensional structure of the protein, like the ionic bonds, hydrophobic interactions, disulfide bonds, and hydrogen bonds. And these forces are involved in the maintaining tertiary structure of the protein, and loss of these forces by some means like the increase in temperature, pressure, pH causes the protein denaturation. So there are so many reagents used for the denaturation of the protein, like guanidinium hydrochloride and urea. You can see here the native state is denatured, is converted into the denatured state by the urea and guanidinium hydrochloride at extremes of the pH, and by increasing the temperature, which causes the aggregation of the protein. So students, let's now summarize what we have learned in this module. We discussed that proteins are synthesized and folded in different conformation, and one of which matching with the native conformation is selected, and that is known as the native state of the protein, which is energetically most favorable state of the protein, and that state is the only functional state of the protein. So protein keeps folding in different correct conformation as it is synthesized in a stepwise fashion. For the quantification of the protein content, it is important, and it has many applications in clinical laboratory practices and research, especially in the field of biochemistry, because if you have any problem, any error in the estimation of protein concentration, that going on in the all process and ultimately lead to the problems in the outcome of the reaction. So that's what it is very essential to correctly estimate the concentration of protein. Destruction of higher order structure leads to the loss of activity of the protein, and this process is referred as denaturation or unfolding of the protein, which occurs mainly because of the loss of all non-covalent forces that holds the three-dimensional structure of the protein. And a small change between the denatured state to the native state, which is equivalent to 1 to 15 kilocalorie per mole, and that's equals to the strength of few hydrogen bonds. So the protein is having mainly native conformation in the cell. It get denatured by extreme of conditions, by loss of non-covalent forces, and that's process known as the denaturation. Thank you.